Hi guys, my name is Emily, and today we're going to talk about the forensic application of organisms and process described in our case study that happened in Switzerland. Okay, so botanical evidence found at the scene. The scene forensic botanist sampled and identified the site's predominant surrounding species and plant litter. All recovered materials were photographed and identified using dichotomous keys and reference collections. Dichotomous keys is a tool created to help scientists identify objects and organisms. It's typically used for identifying a particular type of object or organism that consists of a specific series of questions. And then the reference collections is a collection of objects or organisms maintained for the purpose of study and authentication. So on this slide, I have two examples of dichotomous keys. On the right is one for leaves, and then on the bottom left is one for plants. As you can see, it starts off broad, and then you can kind of narrow it down. So for the plants, it's either it doesn't make seeds or it makes seeds, so it makes it a little easier to understand what you're working with. All right, and for the technique and findings. So the specimens found on the remains were analyzed through the use of the episcope microscope and the optical microscope. The episcope microscope is a critical aspect of observation as it uses fluorescence. This method of illuminating specimens is effective because it reveals features on certain types of plants and everything that you might not be able to see by the naked eye. And then the optical microscope is a microscope using visible light to typically view directly by the light, or by the eye, I'm sorry. So on the left bottom, we have the episcope, and then on the right, we have the optical. So the analysis of bones concluded that the botanical traces were consistent with the species found in the plant litter at the scene.